Welcome to our lecture on steroidogenesis. Today, we are going to explore the fascinating biological assembly line that produces the body's most powerful chemical messengers. These hormones control everything from your response to stress to reproductive development. Understanding exactly how they are made is absolutely fundamental to mastering reproductive biology. To begin, imagine you are walking into a massive, highly efficient manufacturing plant. In this cellular factory, delivery trucks constantly drop off a single raw material, which is a waxy, fat-like substance called cholesterol. The factory workers in our bodies are special proteins called enzymes. These enzymes take the raw cholesterol and start modifying it step by step along an assembly line. Some workers chop off a piece of the molecule, while others add a new chemical tag. Slowly, that basic cholesterol is transformed into powerful chemical messengers like testosterone and estrogen. Now that we understand the factory analogy, let us look at where these factories are actually located in the human body. In formal biological terms, steroidogenesis primarily occurs in the gonads, which include the testes in males and the ovaries in females. It also takes place in the adrenal glands, which sit right on top of your kidneys. At the cellular level, this pathway begins inside the mitochondria. After the initial steps, the intermediate molecules move into the smooth endoplasmic reticulum, where a series of specific enzymatic reactions dictate the final hormone product. Moving deeper into the cell, we must identify the very first step of this assembly line. Inside the mitochondria, a critical enzyme called cytochrome P450 SCC goes to work. This enzyme cleaves the side chain of the cholesterol molecule to produce a new substance called pregnenolone. You can think of pregnenolone as the master starting point, or the universal precursor for all other steroid hormones in the body. Whether the cell eventually makes a stress hormone or a sex steroid, it absolutely must pass through this pregnenolone checkpoint first. Before we dive into the mathematics of this process, let us understand why steroidogenesis is so clinically important. These hormones are the master regulators of human life. Without them, the body could not trigger the physical changes of puberty or maintain a healthy pregnancy. In the real world, medical professionals use their knowledge of this pathway to diagnose conditions. For example, a genetic enzyme deficiency here causes congenital adrenal hyperplasia, leading to severe developmental issues. Furthermore, common medications like birth control pills and fertility treatments work by directly manipulating these exact enzymes and feedback loops. Now that we know what steroidogenesis is, let us look at how fast these enzymes can convert cholesterol into steroids. This brings us to the Michaelis-Menten equation, a fundamental formula from enzyme kinetics. Conceptually, this models the efficiency of our factory workers. V represents the actual rate of hormone production. Vmax stands for the maximum speed when all enzymes are completely busy. S is the concentration of our raw material, cholesterol. Finally, Km is the Michaelis constant, showing the cholesterol level needed to reach half the maximum speed. If cholesterol concentration increases, the production rate increases, eventually leveling off at the absolute ceiling of Vmax. Let us apply this formula to a real biological scenario inside the Leydig cells of the testes, which produce testosterone. We are given a maximum production rate of 120 nanomoles per minute. The Michaelis constant is 40 micromolar, and the current cholesterol substrate concentration is 60 micromolar. We want to find the current rate of testosterone production. We substitute these specific values into our equation. Multiplying the numerator values gives us 7,200. Adding the denominator values gives us 100. Dividing these gives a final rate of 72 nanomoles per minute. This means the enzymes are working efficiently, but can still increase production if more cholesterol arrives. Having calculated the cellular production rate, let us zoom out to measure the total amount of a steroid hormone circulating in a patient's entire body. We use the physiological clearance formula for this. Conceptually, the total production rate of a hormone in the blood must equal the volume of blood cleared of that hormone multiplied by its concentration. P represents the total daily production rate. C stands for the metabolic clearance rate, which is the volume of blood completely cleared of the hormone per day. H represents the steady-state concentration of the hormone in the blood plasma. If either the clearance rate or the plasma concentration increases, the total daily production rate will also increase. Let us see this clearance formula in action to determine if a female patient's ovaries are producing a healthy amount of estrogen. We are given a metabolic clearance rate of 1,350 liters per day. Her blood plasma concentration of estradiol is 400 picograms per liter. We need to find her total daily production rate. We substitute these values into our formula, multiplying 1,350 by 400. This gives us 540,000 picograms per day. To make this manageable for medical charts, we divide by 1 million to convert it to micrograms. The final answer is 0.54 micrograms per day, confirming her ovaries are functioning normally. Now that we have calculated hormone production rates, let us look at the different types of hormones our cellular factory actually makes. Once pregnenolone is created, the assembly line branches into 3 major categories. The first branch produces mineralocorticoids, such as aldosterone, which are essential for regulating salt and water balance in the body. The second branch creates glucocorticoids, like cortisol, which manage your daily physical stress and metabolism. The third and final branch produces the sex steroids, including progesterone, testosterone, and estradiol, which are responsible for sexual development and reproduction. As these hormones move down the different branches, their chemical structure physically shrinks. Steroidogenesis systematically reduces the carbon footprint of the molecules. The raw material, cholesterol, starts as a large 27-carbon molecule. When it becomes pregnenolone, and later progesterone, it is chopped down to 21 carbons. As the pathway continues toward male sex hormones, testosterone is formed, containing just 19 carbons. Finally, a specialized enzyme called aromatase converts testosterone into estradiol, which is the primary female sex hormone, leaving it with only 18 carbons. This precise chopping is what gives each hormone its unique function. Let us bring all of these steps together in this comprehensive pathway diagram. At the very top, you see our raw material, 27-carbon cholesterol. Notice the critical first arrow labeled P450SCC, which creates the universal precursor, pregnenolone. From there, the flowchart clearly splits into our 3 main branches. On the left, we have the mineralocorticoids, ending in aldosterone. In the center, the glucocorticoids, ending in cortisol. On the right, follow the sex steroids pathway, down through progesterone and testosterone. Finally, observe the very last step at the bottom right, where the enzyme aromatase converts 19-carbon testosterone into 18-carbon estradiol. We have covered a lot of ground today, so let us review the key takeaways. First, steroidogenesis is the vital biological manufacturing process that converts basic cholesterol into life-sustaining steroid hormones. Second, remember that the enzyme cytochrome P450 SCC is the gatekeeper, creating the universal precursor, pregnenolone. Third, we can mathematically predict how fast these cellular factories work using the Michaelis-Menten equation, and we can measure total circulating hormones using clearance formulas. By understanding both the biological steps and the mathematical rates, you now have a solid grasp of how our bodies maintain internal balance and drive reproduction. Great job today!